Can somebody confirm you can hear me?
I can hear you.
Thank you. All right, before we begin, are there any questions? If there's no questions, let's begin. Today is May 8th, and we're supposed to be discussing Lab 7, the throat and urine cultures. There really is no unknown work to do because I haven't given you your number or your results. I'll send those out on Tuesday, but you could, if you wanted to, make a note that you are taking your culture and pretending to streak it out. Any question about any of that? All right, you've already started the glucose hydrolysis project. You started that on Tuesday. And on Tuesday, before I send you the results, you'll be starting the gram stain. So lab 7, throat and urine cultures. Actually, I think I'll close that down because my computer's a little slow at the moment. And it's still working on the lecture file. That's why it's slow. So lab 7, throat and urine cultures. As is normal, there is some reading in the textbook, but not very much this time. You can read page 392 about nose, throat, and mouth normal microbiota. You can read about the beta hemolytic streptococci, which there's a section at the end of this lab module on that, as well as Haemophilus. There's a section at the end of this lab module on Haemophilus too. You can read about the oxidase test on page 132 and the catalase test on page 155. The coagulase test, I've added something to this document in the appendix so you can read the lab module. There's some video clips in this material, this lab. The virtual lab had to be removed because it used, what was that called? Flash, which is no longer supported. The learning objectives for this lab: first, be able to explain how to properly collect throat and urine specimens for routine culture. Two, list the common bacterial pathogens isolated from throat and urine cultures. Three, know the culture media used to cultivate these pathogens. Four, recognize which tests are used to identify throat and urine pathogens. Five, understand the principles and how to interpret the following tests: the catalase test, the coagulase test, and the oxidase test. We will talk a little bit about the XV factor testing, but you do not need to know about that. I won't quiz you on that. Introduction. Once specimens are received in a lab, they are logged and processed in a timely fashion. Specific culture media are inoculated and incubated depending on what the clinician or the lab thinks it is, depending if it's a throat or urine specimen. Below is an overview of the culture media used to cultivate common bacterial pathogens isolated from throat and urine cultures. For the throat, there's really only two media you need to know, and that's blood agar and chocolate agar. Streptococcus pyogenes is the most common bacterial pathogen isolated from the throat. However, in infants and young children, Haemophilus influenzae can also be isolated, and Haemophilus influenzae cannot grow on blood agar, so we also use chocolate agar, especially when you're taking a throat culture from a infant or a young child. For urine cultures, we always use blood agar, and we use either McConkey's agar or EMB agar, EMB, eosine methylene blue agar, to grow our pathogens. Streptococcus, Enterococcus, and Staphylococcus all grow on blood agar, as well as Enterobacteriaceae and Pseudomonaceae, all grow on blood agar. But most of the, or I should say the majority of the pathogens are either in the family Enterobacteriaceae or Pseudomonaceae, and these are gram-negatives. And gram-negatives grow on MacConkey's and EMB. Gram-positives do not. And so that's why we will grow or culture, if it's a urine pathogen, always on two media, blood agar, and then either MacConkey's agar or EMB. Any question about any of that? All right. Like if you streak the throat of an adult, you only use blood agar. If you streak the throat of a young child or an infant, you'd use chocolate agar. I'm not sure if you would also use blood agar. I think you would also use blood agar, and there's a reason for that, and that is um, Streptococcus pyogenes will make a specific showing on blood agar, so you'd probably streak it on both. Haemophilus influenzae for the chocolate agar and the Streptococcus pyogenes for the blood agar. The Streptococcus pyogenes would grow on the chocolate agar, but it would not give you the beta hemolysis on the chocolate agar, so you'd have to grow it on blood agar. And that's not something you need to know about. But throat cultures are largely due to viruses and bacteria. A virus won't subculture on in an agar plate, but if you see white patches on the tonsils or in the back of the throat, it's indicative of a bacterial infection. If it's only redness in the throat, then it's likely a viral infection. Now, the bacterial infection will also be red in the throat, but you get the white patches. The most common isolated bacterial pathogen is Streptococcus pyogenes, also known as Group A Streptococcus or gas. I don't use that one, and I almost never use Group A Streptococcus. I use the species name. Strep throat is caused by Streptococcus pyogenes, and you swab the back of the throat or the tonsils where there's redness and or white patches, avoiding touching the tongue, the sides of the mouth, and the roof of the mouth. A tongue depressor will help with that. Patients that test positive for Streptococcus pyogenes are usually placed on antibiotic therapy. The reason being that Streptococcus pyogenes has a capsule around it, which helps prevent the immune system from at least easily attacking and removing the Streptococcus pyogenes from the body. So many patients, I was one, once you have a Streptococcus pyogenes invasion, it's not thrown off by the body. Some patients can throw it off, but generally it's not thrown off, and then will develop into strep throat. And then if it goes better, it can develop scarlet fever and a bunch of other diseases. I've forgotten their name because they're not very common anymore. But before antibiotics, scarlet fever was very common disease, and all the other diseases that strep throat could have developed into. Streptococcus pyogenes, or gas, or Group A Streptococcus, is a fastidious organism, meaning it will not culture on a general purpose media. So you have to grow it on a enriched media, such as blood agar, where the blood cells supply the nutrients that Streptococcus pyogenes needs before it'll grow. You then examine the blood agar plate for small white colonies that have complete zones of hemolysis. Let me blow this up way high. So right here, we have a normal throat culture where if you look at the agar up until the colony, it's completely red. So there's no hemolysis. Right there, we have a little bit of alpha hemolysis. And right there, we also have alpha hemolysis, which is a darkening of the blood, but it's not complete hemolysis. And so this would not be Streptococcus pyogenes, and that's not Streptococcus pyogenes either. None of these colonies are Streptococcus pyogenes. Excuse me. Sorry about that. I had to sneeze. This plate is showing you a small white colony right there in the center, surrounded by a much larger clear zone where the blood cells have been lysed, or complete beta hemolysis. That colony there doesn't have beta hemolysis. That colony actually has alpha hemolysis. You can see a dark ring surrounding the white or off-white colony. But right there, we have a small white colony, and that's Streptococcus pyogenes having complete hemolytic activity. And you can see most of these colonies here have beta hemolysis. So do these colonies there. Any questions about any of that? All right, let me shrink this down. All right, there are two tests that are needed to differentiate between beta hemolytic bacteria and Streptococcus pyogenes. So not all beta hemolytic colonies on a blood agar are caused by Streptococcus pyogenes. There are other beta hemolytic bacteria, including beta hemolytic Streptococcus. But there are only two tests you need to show that the beta hemolysis is caused by Streptococcus pyogenes. One is running the catalase test. Catalase is an enzyme that breaks down hydrogen peroxide. So here we have hydrogen peroxide around catalase will produce water and oxygen. And so if you see bubbling in the hydrogen peroxide, you know the enzyme catalase is around. If the enzyme is positive for catalase, bubbles will be shown. And the majority of bacteria that grow in air are catalase positive. However, the families of Streptococcus and Enterococcus are negative for catalase production. But all other aerobes, or most other aerobes, I should word it that way, most other aerobes are negative for catalase. Most other aerobes are positive for catalase production. The second test you need to show that a beta hemolysis colony on blood agar is Streptococcus pyogenes is by running the bacitracin test. So catalase, if it's catalase negative, it's either a member of Streptococcus or Enterococcus, but there are other species besides Streptococcus in those two families. So you do the bacitracin test. Bacitracin is an antibiotic, so you coat the area of the plate with the beta hemolytic bacteria, and then put on the basotracer disc, and then you grow the culture. And if the bacteria are sensitive to basotracin, then it is a basotracin positive test. And it's Streptococcus pyogenes. If, on the other hand, the bacteria is not sensitive to basotracin, it is negative for the basotracin test, and it is not Streptococcus pyogenes. Any question about any of that? All right, so if it forms beta hemolysis on blood agar, you run the catalase test. If it's catalase positive, it's not Streptococcus pyogenes. And if it's catalase negative, you didn't run the basotracin test. And if it's basotracin positive, it is Streptococcus pyogenes. If it's basotracin negative, then it's some other species, another member of Streptococcus or Enterococcus. That is beta hemolytic. All right, another organism that can be clinically significant when isolated from the throat is Haemophilus influenzae. It is a gram-negative coccobacillus. Haemophilus influenzae is a common cause of ear, eye, and sinus infections in babies and young children. Haemophilus influenzae is fastidious. However, it cannot grow on blood agar because Haemophilus influenzae does not lyse the red blood cells. And even though the red blood cells do provide what Haemophilus influenzae requires, Haemophilus influenzae will not release the requirement because it cannot lyse the red blood cells. Streptococcus pyogenes, by the way, can because it does lyse the red blood cells. So we grow Haemophilus influenzae or a culture from the throat on a chocolate agar, and then Haemophilus influenzae will grow as shown here on chocolate agar. Remember, chocolate agar is blood agar, but instead of adding the blood after the agar has cooled to something like 50 degrees Celsius or lower, you add the blood immediately when you pull the agar out of the incubator, and that will lyse the blood, causing the agar to take on the brown appearance, making it look like you've added chocolate to the agar. By lysing the red blood cells, you will release the ingredients from the red blood cells that are required by Haemophilus influenzae, and so they'll grow on chocolate agar. They will not grow on blood agar unless you do something like put a streak of Staphylococcus aureus down the blood agar plate, and then you put the Haemophilus influenzae streak it perpendicular to the Staphylococcus aureus. And then the Staphylococcus aureus will lyse the blood agar. In fact, if I blow it way, way up, you can actually see it. There we go. Hopefully you can see that now. See that thin white line running down the middle of this streak? That is from the streak of Staphylococcus aureus. And then the clearness, the white region around that streak, is from the complete hemolysis of the red blood cells around the Staphylococcus aureus. The Streptococcus, around the Staphylococcus aureus, the Haemophilus influenzae, which is streaked this way, will not grow in this region of the plate but it will grow in this region of the plate because the Staphylococcus aureus has lysed the red blood cells here, and then the nutrients from the red blood cells have diffused into the plate to here to here. But after that mark, the nutrients do not diffuse far enough out, and then after that point, Haemophilus influenzae will not grow. Any question about either of these plates? You probably don't need to know about this plate. I don't think I'm going to ask you any quiz questions about a blood agar plate having a Staphylococcus aureus streak down the middle of the plate, and then the Haemophilus influenzae streak perpendicular to the Staphylococcus aureus, allowing the Haemophilus influenzae to grow near the Staphylococcus aureus, but not growing further away. But you should know that Haemophilus influenzae can grow on a chocolate agar plate because it does supply the factors required for Haemophilus influenzae to grow. And the factors that it require are the X and the V growth factors. All right, any questions about throat cultures? If not, let's talk about urine cultures. Urine cultures are used to diagnose urinary tract infections. Bacteria can travel up the urethra to the bladder. And then if not cured, it can even travel up the bladder to the ureters, up to the kidneys. That doesn't usually happen, but it can happen where you get a very serious urinary tract infection that goes all the way up to the kidneys. Females are more predisposed than males in developing UTIs because females have a shorter urethra. To sample for a UTI, patients can be sampled in one of three ways. The way that the patient usually likes is the clean catch midstream, where the genital area is clean prior to collection, and then you collect the urine midway through the urine, so not at the beginning of the urine flow, not at the end of the urine flow. And that will reduce the contaminants from the urethra getting in the urine. If you wanted to clean up the urine even cleaner, you can also put a catheter directly into the bladder and then sample the urine that way. The trouble is the catheter is going through the urethra. The catheter can pick up bacteria from the urethra and then bring the bacteria into the urine before it's used by the catheter. And then the best way is to suprapubic aspiration, where you insert a needle through the abdominal wall directly into the bladder and sample the urine directly from the bladder. And that way should not give bacteria into the urine. The patient doesn't like this procedure because obviously it's using a needle stuck through their intestine going into the bladder. The patients don't really like catheters either, but they'd probably prefer that to the suprapubic aspiration. Anyways, that's three ways you can collect urine. Once the specimen is then received in the lab, 0.001 millimeter, that's 1/1000th of a milliliter, or 1 microliter of urine is placed onto a blood agar plate using a special inoculating loop that measures exactly one microliter. And there's the inoculating loop there, shown next to a penny. Canadian penny, but a penny. And then what you do is you streak the urine on that pattern and that way too. Look at the growth on the blood auger plate, and then take the urine probably, and then streak it in either a McConkey's plate or an EMB plate. You streak it on McConkey's or EMB because the majority of urinary tract infections are caused by a gram negative, either a member of family Enterobacteriaceae or family Pseudomonaceae, and these species will grow on McConkey's and EMB plates. They'll also grow on blood auger plate, but the point is you remove some of the possible contaminants if you expect that it's a gram negative infection by growing on McConkey's or EMB. But not all urinary tract infections are caused by gram negatives. Some are caused by gram positives, like Staphylococcus aureus, and it won't grow on McConkey's or EMB, but it will grow on blood auger. The interpretation of a positive urine culture is multifaceted. Some factors that need to be considered is the type of collection. Voided, even the clean catch voided method, will have more contaminants than the catheterized specimen or the suprapubic aspiration specimen. The gender of the patient, males have fewer contaminants than females. The number of colonies per mil per urine, and then the presence of microbial growth. Like if there's multiple organisms isolated, it may indicate a poor collection technique. Any questions about any of that? Some tests you do for the urine specimen is you can do the coagulase test. Coagulase looks for the production of the enzyme coagulase. This test is used to differentiate Staphylococcus aureus from other members of the Staphylococcus family. Staphylococcus aureus is coagulase positive. The other members of the Staphylococcus family are all coagulase negative. Coagulase production actually contributes to the pathogenicity of Staphylococcus aureus, converting blood plasma into fibrin, meaning a blood clot. And the clot helps protect the bacteria from the immune system, particularly when the bacteria numbers are small, and would be easiest for the immune system to remove the bacteria from the body. So the clot from the enzyme coagulase helps protect the bacteria from phagocytosis by the immune system cells. And what happens is fibrinogen in the plasma will get together with coagulase to make the fibrin clot, and the fibrin is the fibrinogen after the coagulase, which will then begin clotting the blood. There are two tests to look for coagulase. There's the slide test, where you put the bacteria in the plasma, and if the bacteria produce coagulase, you'll get clumping of the bacteria in the plasma. If there is no coagulase, the bacteria will look like this, no clumping. You can also do the tube test, where if you are negative for the enzyme coagulase, the plasma will stay a liquid, and you tilt the tube, it'll look like that. And if the coagulase is present, the plasma will clot, you'll get a clump like that. Any question about the coagulase test? All right, the most common bacteria isolated from a routine UTI culture is E. coli. a member of the family Enterobacteriaceae, and there are other members of the family Enterobacteriaceae which can cause a UTI, making the UTIs being caused by the family Enterobacteriaceae, I believe a little bit over 50%. And then if you include the family Pseudomonaceae, then the gram negatives make up around 60%, maybe a little bit over 60% of the UTIs. Any question about anything so far? All right. Another enzyme we can use to test the urinary tract infection is the oxidase test. Oxidase looks at the cytochrome C oxidase, and if the organism performs aerobic respiration, it has cytochrome C in it, which obviously will make the oxidase test positive. The family Pseudomonaceae, such as Pseudomonas aeruginosa, is oxidase positive, and then all the members of the family Enterobacteriaceae are oxidase negative. So the oxidase test is frequently used to distinguish an infection that's caused by the family Enterobacteriaceae from the family Pseudomonaceae. So if you have a urinary tract infection which grows on McConkey's plate or EMB, then we do the oxidase test to determine whether it's from the family Enterobacteriaceae or the family Pseudomonaceae. There are other ways to distinguish the family Enterobacteriaceae from the family Pseudomonaceae. The family Pseudomonaceae are all aerobic, non-glucose fermenting bacteria. The family Enterobacteriaceae are all facultative anaerobes, so they're not aerobes, and they all ferment the sugar glucose. But the oxidase test is the one frequently used to distinguish it, probably because the oxidase test is a very quick test and you get the results right away. But you could look for growth in a how do you say that word? Glycoculase? That's not right. I forget the name. Glycoculase? Glycoculate? I don't remember the term. Not saying it correctly, but something like a glycoculase media. And look at that. And you could also ferment the bacteria with glucose to distinguish the two families. The oxidase test is frequently used to differentiate Enterobacteriaceae from Pseudomonaceae because it's very quick, and you just put the bacteria on and the oxidase, and if the oxidase is present, it will turn blue. If oxidase is absent, you will not see the blue within 30 seconds. All right. Urine and throat cultures are two of the more common types of specimens received in a lab for bacterial cultures. Microbiologists are trained to isolate and identify present pathogens that are present. You can read about the references. Actually, I would ignore them, but that's just me. You're not going to be performing any of these activities. Everything will be done online, but you will be held responsible for knowing how to perform them and how to interpret them. Here is a video on the catalase test, so watch that video. There's a video on the coagulase test, so watch these videos, and these are very short videos. And then watch this video on the oxidase test. It's also a very short video. All right, let's go to the worksheet now. And then answer these questions. Answer provided by an AI may be marked wrong. For questions 1 to 3, identify the principle used to detect the presence of catalase, coagulase, or oxidase. So questions 1, 2, and 3 use one of these three enzymes that we talked about in this lab. And 1A, what enzyme converts fibrinogen into fibrin? And then 1B, name a microbe that tests positive for this enzyme. If you give an enzyme, which is not mentioned in this lab, that you got from AI, like I stated, it may be marked wrong. So you must use something we've talked about in this lab. And then 2, which enzyme detects the presence of cytochrome C proteins in the electron transport system? And then 2, name 2 microbes that test negative for this enzyme. So that'd be 1A, or excuse me, 2A, and this would be 2B. And then 3A, what enzyme hydrolyzes hydrogen peroxide into the non-toxic products, water and oxygen? And then 3B, name 2 microbes that test negative for this enzyme. And 4, which enzyme is used to differentiate between the following? Staphylococcus and Streptococcus, one of the enzymes we mentioned above. And then which enzyme is used to differentiate Staphylococcus aureus from other Staphylococcus species? Name that here. And then 4C, which enzyme is used to separate Enterobacteriaceae from Pseudomonaceae? 5, concerning specimen collection for a routine throat culture, what areas of the throat should be swabbed? So there's 2 reasons there, or 2 areas there. And then what areas of the mouth should be avoided? And list those there. 6, what is the most common species causing bacterial throat infections? Mention that there. And 7, what test is used by microbiologists to differentiate beta hemolytic group A Streptococcus, meaning Streptococcus pyogenes, from other beta hemolytic Streptococcus species isolated in a throat culture? So mention the test or the enzyme here. And note, we already know that this is a beta hemolytic. Bacteria. And we're only separating family Streptococcus from Streptococcus pyogenes from other beta hemolytic Streptococcus species. 8, answer the following concerning a child's throat culture. State 2 non-selective agar plates that are inoculated. What 2 medias can grow a child's throat culture? And then what organism can be isolated on CAP but not on BAP? 9, list 2 media that are used to grow urine cultures. And then 10, list 3 reasons why urine specimens should be rejected for culture. You can get the answer to that in the appendix. 11, how much urine is placed on the surface of a blood agar plate? 12, what is the most common bacteria isolated from patients with a bladder infection? And you should name the species. 13, identify the Gram reaction, what the Gram stain is, and the cell morphology the shape, not the growth arrangement of these bacteria. Scientific name for shape, cocci, not balls or not spheres. And if you can't find the answer, you should be able to find the answer in this lab, by the way. So give me the gram stain of Pseudomonas aeruginosa and the shape, the gram stain of E. coli and its shape, the gram stain of Staphylococcus aureus and its shape, the gram stain of Streptococcus pyogenes and its shape, and then the gram stain of Haemophilus influenzae and its shape. Any question about any of that? All right, get working on the lab. This will be your last easy lab. The other labs will take you a little longer. I will be here until 8 o'clock, or the last student logs out to help you with any questions on the lab. You question early because once the last student logs out, I will log out as well. All right, get started on the lab.